Greetings to all. In this lecture, we are going to see about cells of immune system. Hematopoiesis is the process by which all cellular components of blood are formed and developed. This continuous process is essential for maintaining a steady supply of blood cells, including red blood cells and white blood cells. Hematopoietic stem cells are the cells from which the cells are originated from. All the blood cells originate from a small population of hematopoietic stem cells. These hematopoietic stem cells are multipotent, that means they have the ability to differentiate into all the various types of blood cells. They also have the capacity for self-renewal, ensuring a continuous supply of stem cells throughout an individual life. Hemopoietic stem cells reside primarily in the bone marrow in adults. The primary site of blood cell production changes throughout the life. Yolk sac during early embryonic development, liver and spleen during fetal development, bone marrow during late fetal development and adulthood. By the end of the gestation, the bone marrow becomes the primary and lifelong site of hematopoiesis. In adults, active red bone marrow is mainly found in the axial skeleton and the proximal end of long bones. Under certain pathological conditions, hematopoiesis can occur outside the bone marrow in organs like the liver and spleen, reverting to fetal sites of blood cell production. Hematopoiesis is a hierarchical process involving several stages of differentiation and maturation. Hematopoietic stem cells, the most primitive cell with cell renewal and multipotential ability, differentiate into multipotent progenitors, which have lost some several renewal capacity but can still give rise to multiple lineages. The multipotent progenitors diverge into common myeloid progenitor, which are the precursor for myeloid cells, such as granulocytes, monocytes, macrophages, erythrocytes, and platelets. Common lymphoid progenitor, which are the precursor for lymphoid cells, such as B cells, T cells, and NK cells. The common myeloid progenitor and common lymphoid progenitor further diverge into lineage-specific progenitors. These progenitors mature into recognizable precursor cells such as erythroblasts, myeloblasts, and immunoblast cells. The mature blood cells are differentiated from the precursor cells, and the maturation become functional, and the mature blood cells that circulate in the bloodstream later. Now we are moving on to the myeloid lineage cells. We are going to see about the structure of neutrophils. The neutrophils are the most abundant type of white blood cells, making up to 50 to 70% of circulating leukocytes in humans. They are characterized by their size, nucleus, cytoplasm, and organelles, and their own mobility. They are relatively uniform in size with a diameter of 9 to 15 micrometer. Their most distinctive feature is a multi-lobed nucleus, typically consisting of 3 to 5 segments connected by a thin strands of chromatin. This gives them the name polymorphonuclear leukocytes. As neutrophils mature, the nucleus becomes more segmented. The cytoplasm contains numerous fine granules that stain neutrally, pink or purple blue in color, with Romanowski dyes, hence the name neutrophil. These granules are membrane-bound vesicles containing a variety of antimicrobial proteins and enzymes. Neutrophils have a relatively small number of other organelles such as mitochondria and ribosomes, which reflects their short lifespan. They exhibit amoeboid movement, allowing them to migrate out of the bloodstream and into the tissues. They can extend pseudopods to move along surfaces. Neutrophils are crucial components of the innate immune system and play a vital role in the body's defense against infection and injury. Their primary functions include phagocytosis. They can engulf and destroy pathogens such as bacteria and fungi, foreign particles, and cellular debris. They release antimicrobial substances. Neutrophils can release the content of their granules into extracellular environment to directly kill pathogens that are too large to phagocytize or to amplify the inflammatory response. This process is called degranulation. Neutrophil extracellular trap formation is also their ability. In response to certain pathogens or inflammatory stimuli, neutrophils can release a web-like structure composed of DNA, histones, and granular proteins. These net can trap and kill extracellular microbes, preventing their spread. This process is called netosis. Neutrophils play a complex role in inflammation. They are among the first immune cells to arrive at the site of injury and release various mediators, including cytokines and chemokines, that contribute to the inflammatory response by recruiting other immune cells to the site, increasing vascular permeability, contributing to pain and swelling, whilst are crucial for initial defense. Uncontrolled neutrophil activity can also contribute to tissue damage. The next cell is eosinophil. Eosinophils are a type of granulocytes, a category of white blood cells characterized by the presence of prominent granules in their cytoplasm. Eosinophils are typically larger than neutrophils, ranging from 12 to 17 micrometer in diameter. They are spherical in suspension but can become more amoeboid when migrating into tissues. Their nucleus is typically bilobed, meaning it has two distinct segments connected by a thin strand of chromatin. This is a key characteristic that helps distinguish them from the other granulocytes. The cytoplasm is packed with large eosinophilic granules. These granules are membrane-bound organelles that stain intensely red or pink with the acidic dye eosin in the Ramanowski staining method. Eosinophils contain around 200 large granules, which are their defining feature. These granules are not uniform and can be categorized into crystalloid core, granular matrix. Compared to other cells, eosinophils have a relatively modest amount of other granules like mitochondria and endoplasmic reticulum, reflecting their terminally differentiated state and shorter lifespan. Eosinophils' cell surface expresses a variety of receptors that allow them to interact with their environment and respond to various stimuli. These include receptors for cytokines, chemokines, immunoglobulins, complement components, and lipid mediators. Eosinophils play a complex role in the immune system, primarily involved in defense against parasitic infections. This is considered their classical and most crucial role. Eosinophils are particularly effective against helminth parasites that are too large to be phagocytized. They achieve this by adherence, degranulation, and oxidative burst. Eosinophils are key players in the pathogenesis of allergic diseases, contributing to both the initiation and resolution phases, such as inflammatory mediator release and cytokines. The next cell is basophils. The basophils are the least common type of granulocytes, making up only about 0.5 to 1% of circulating white blood cells. They share some structural similarities with other granulocytes but have unique features. Basophils are relatively large, typically ranging from 12 to 50 micrometers in diameter. They are spherical in suspension. They possess a bilobed or S-shaped nucleus, which is often obscured by the numerous granules in the cytoplasm. The chromatin is also condensed. The cytoplasm is plaqued with large dark blue to purple black staining granules when using basic dyes like hematoxylin in the Ramanowski staining method. These basophilic granules are their most distinguished characteristic. These granules are membrane-bound vesicles containing a variety of potent bioactive substances such as histamine, heparin, chondroitin sulfate, proteolytic enzymes, cytokines, and platelet activating factor. Despite their low numbers in circulation, basophils play an important role in various physiological and pathological processes. such as allergic reactions and hypersensitivity. And this is achieved through vasodilation, increased vascular permeability due to histamine, leakage and edema, smooth muscle contraction, mucus production, and itching. Similar to basophils are thought to play a role in defense against certain parasites, particularly helminth worms. Basophils can present antigens to T cells also, further influencing the adaptive immune response. The release of heparin from basophil granules contribute to local blood anticoagulation, which may be important in certain inflammatory or infectious settings to maintain blood flow. The next cell is monocyte. Monocytes circulate for about 1 to 3 days in blood after formation before migrating into tissues throughout the body. A key characteristic of monocyte is their ability to differentiate into other important immune cells once they have the bloodstream and enter into other tissues. The two main types of cells they can become are macrophages and dendritic cells. The monocytes are the largest leukocytes, typically 12 to 20 micrometer in diameter, about twice the size of red blood cells. They have a large single kidney-shaped horseshoe-shaped nucleus. This mononuclear characteristic distinguishes them from polymorphonuclear leukocytes that have multi-lobed nuclei. Their cytoplasm is relatively abundant and appear grayish-blue when stained. Monocytes are their differentiated forms, such as macrophages and dendritic cells, perform a wide range of crucial functions in immune system. Their ability to differentiate into macrophages and dendritic cells equip them with a diverse function essential for host defense, tissue homeostasis, and immune regulations. The next type of cell is the macrophages. Macrophages are a type of white blood cells that reside in tissues and play a crucial role in the immune system. Derived from monocytes, macrophages are large, specialized cells that are essential for phagocytosis, antigen presentation, and cytokine production. Macrophages are professional phagocytes, meaning they engulf and digest cellular debris, pathogens such as bacteria, virus, fungi, and parasites, foreign substances, and even tumor cells. This process is a primary way they eliminate threats and maintain tissue homeostasis. They act as an antigen presenting cells, particularly when they differentiate into dendritic like cells. They engulf and processing pathogens. They display fragments of these pathogens on their surface along with major histocompatibility complex molecules. This presentation is crucial for activating other immune cells, especially T cells, and initiating the adaptive immune response. The macrophages produce a wide array of cytokines and chemokines, which are the signaling molecules that recruit other immune cells to initiate the site of infection or inflammation, regulate the inflammatory response, and influence the activity of other immune cells, including lymphocytes. The next type of cell is dendritic cells. Dendritic cells are specialized type of antigen presenting cells that play a crucial role in the initiation of adaptive immune response. They act as the primary link between the innate and adaptive immune systems by capturing antigens in peripheral tissues, migrating to secondary lymphoid organs like lymph nodes, and presenting these antigens to T lymphocytes, thereby activating them. Dendritic cells are characterized by their distinct dendritic branch-like processes or progesins, which increase their surface area for antigen capture and interaction with immune cells. This morphology gives them their name, resembling the dendritic of nerve cells. They are strategically located throughout the body, particularly in peripheral tissues, secondary lymphoid organs, and in bloodstream. The primary function is the antigen presentation to T cells, leading to the initiation of adaptive immunity. They are highly efficient at this process. Antigen presentation to the T cells are affected by the dendritic cells. In the lymph nodes, mature dendritic cells present the processed antigen peptides bound to major histocompatibility molecule to native T cells. But thus, we have come to the conclusion of the lecture. We'll see you in the next lecture. Thank you.